Good morning. I'm delighted with the turnout and the interest in this, we think, really very important topic. I'm Stan Gerson, the director of the Case Comprehensive Cancer Center, and the forum that we're going to be talking about today is the impact of genomics on cancer diagnosis and treatment, especially with some reference to issues that it raises in terms of policy. And I mean by that, and I think as we'll hear today, both institutional policy, clinical trials and clinical trial policy, reimbursement policy, FDA policies, issues that really we all confront a lot of our time, both in the research and clinical missions that we all participate in. We've got a very exciting day, and we're going to be busy, so I'm going to try to make sure that we stay on time. And after a rules presentation during our moderator sessions, the moderators will see a sign go up 10 minutes before the end of their time, asking for questions from the audience so that we have plenty of time. Since most of our discussion today will be a panel, which we'll put up after our first presentation. In fact, these seats down here in the front, which are often blocked by the speaker, can be filled. And we're going to, I think, be pretty full during the day. So what do we want to accomplish during the day today? One is to review the status of clinical genetic testing for precision drug selection for cancer patients, discuss clinical, regulatory, and research questions related to implementing cancer genomic testing in the clinical space at cancer centers and individually by physicians, and to think about the issues that might let us reach a consensus on how to optimally develop a national approach and policy on improving patient care and outcomes through cancer genomic testing. That may sound a little bit ambitious, but I think if you give it some thought, we really don't want to be in a situation in which we're individually trying to manage this over time. What are the origins of this conference? Well, I was really behind the times on this until it came up at the NCI directors and Board of Scientific Advisory meetings for the NCI, where we had a series of presentations on precision medicine, and it went very quickly from highfalutin science, in which we were delighted to see all those fancy things that those molecular biologists could do, to realizing that there were actually some clinical decisions to be made. And we began having some discussions with other cancer centers about how you actually implement this. It was active at a few centers in the research space. And the Case Comprehensive Cancer Center, its clinical partners, at university hospitals and Cleveland Clinic, then began to think about how would they actually do, test, bill, implement clinical genomics. And we had the recognition that decision-making was actually pretty complex, that reimbursement was either non-existent or uneven, raising the scenario that, and we've certainly seen examples of this, of one-offs, of a patient who would actually, outside of their insurance carrier, need to be able to pay for testing, and the use of an unapproved indication for, or an unapproved drug that was indicated based on the genomic testing. And that's a system that just didn't make cognitive sense to any of us. So we have on the panel members from 6 different NCI-designated cancer centers, representatives from the NCI, FDA, CMS through video conferencing, 200 or so of all of you. And all of you are physicians, genomic investigators, commercial partners, developers, support personnel, and some patient advocates. I'm delighted that it's not common, if you will, for newspapers to want to report on a conference. Like, is there news there? But we did, in fact, make it into the Plain Dealer today. And we'll get copies of this so that you can sort of see that we were quoted, and that they actually mentioned a patient who will be here around noon to talk about his own individual experience. So logistics, and then I'll get off the plate for you all. Breaks and lunch are outside where you all signed in. Notes will be taken because we actually want to bring some of this information forward and put together a white paper from our discussion. I will certainly appreciate comments and substantive issues that you see with our discussion today by email, which is stanton.gerson@case.edu. So That would be very helpful since we want to try to get to some consensus. We will be having a social media tweet, not that I know how to do that, but it's right there for you all. And I especially want to thank the incredible effort of our team, Jillian Todd, Ann Dooley, Chris Samosi, Chris Sheridan, and the support that we've received from the chair of our Cancer Council for the Comprehensive Cancer Center, Jerry McDonough, who gave us some funds to help get this off the ground. So I'm going to move directly on to introducing a rule. Arul Shanaian is a clinical pathologist, has taken the lead at the University of Michigan in understanding molecular genomics and the therapeutic decision-making that's required. He's a Howard Hughes Medical Institute member and is the S.B. Hicks Endowed Professor of Pathology and Pathology of Urology at the University of Michigan, where he's been for a good number of years. He is, in fact, the inaugural director of the University of Michigan Center for Translational Pathology and really brings together all the omics capabilities. He's really thought about this more than any of us, I think. Has well over 250 publications, usual membership for highfalutin individuals, Association of American Physicians, and a few others, has received a number of awards. Critical, I think, is his paper about 2.5 years ago now in Science Translational Medicine, which he identified an approach and a strategy for actually taking the omics information and bringing it to therapeutic decision making. And we thought it would be terrific to have Arul help kickstart our discussion for today. Thank you.
Thank you, Stan, for the opportunity to kick off this session. It's a very important topic. I think it's certainly an exciting time to be in cancer research and cancer therapy, primarily because I believe that the technologies have matured to the point where we can now comprehensively map the mutational landscape of an individual patient, compare that to the normal genome, and pretty much get at the molecular basis of an individual patient's tumor. And of course, by fully understanding the molecular basis of an individual's tumor, the hope is that we will be able to better or more precisely target that individual patient's aberrations or pathways that might be affected. However, I think there are a number of challenges to actually bring this type of technology into common utility, a number of hurdles in terms of approval processes, regulatory processes, and of course reimbursement issues that we actually need to achieve in order to actually make this type of technology more useful to essentially scale this technology in terms of utility. And so what I thought I would share with you is sort of an abbreviated talk on our experience at the University of Michigan in setting up a clinical sequencing effort, essentially initially as a pilot feasibility approach, but with the idea that we would gain information in terms of what are the challenges as we move forward. And we initiated this project, or we enrolled our first patients in 2011. And since then, we've now sequenced over 200 adult patients. We've also established a parallel pediatric oncology effort in which we've enrolled over 30 individuals. So it's beginning to gain traction, and we're beginning to sort of understand some of the issues as we move forward. But I think it's great that Stan has brought together diverse expertise at this meeting to sort of begin to focus on the actual issues of implementation. Just a few disclosures that I need to mention. I'm involved in a variety of capacities with different companies that are involved in some aspect of sequencing, including Life Technologies, Paradigm, which is a nonprofit spin-out from the University of Michigan. You might hear a little bit about this from Bob Penny, who's joined us, and I'm part of their SAB, Hologic, as well as Molecular MD. So the manuscript that Stan was referring to is this manuscript that featured the sequencing of the first 2 patients that we enrolled in our IRB-approved protocol. We called this project MyOncoSeq for the Michigan Oncology Sequencing Program, where we're essentially evaluating the potential merits and challenges of comprehensive sequencing-based analysis for cancer patients. And this really stems from a number of consensus reports suggesting the importance of this and the fact that the technologies have now matured to the point where we can now comprehensively understand the molecular basis of human disease. For example, this consensus report by the National Academy of Sciences, which was published in late 2011, really pushing us towards precision medicine, or that the time is right to develop the molecular taxonomy of human disease, especially in the context of cancer. And there have been a number of reasons for this, certainly advances in sequencing-based technologies, but this is just probably a slide that many of you have seen, in that the cost of sequencing has decreased markedly, faster than Moore's law, which is generally associated with the cost of semiconductors in the computer industry. The cost of sequencing has precipitously decreased relative to Moore's law. It took the first draft of the human genome was completed in about 2000. Took over a decade to complete and over $3 billion. We can now do a similar type analysis for several $1,000s within several weeks. So now the technology is at the point where one could consider that every cancer patient should get some sort of genomic assessment carried out. And here I'm just going to share with you a few examples that many of you, I'm certain, are aware of why it's important to understand the mutational landscape of an individual cancer patient. This is the BRAF V600E mutation in malignant melanoma. This patient is riddled with tumors here and treated with the targeted therapy vemurafenib. You can see that the tumor, at least initially, melts away in this particular patient. And these are patients that have the V600E mutation that respond to this drug, this targeted therapy. A similar example in the context of a gene fusion, the EML4 ALK fusion in non-small cell lung cancer. This patient is afflicted with a fusion-based, an ALK fusion-based cancer, and is treated with the small molecule inhibitor crizotinib. And again, the tumor melts away in this context if this patient has this particular mutation or fusion, is matched with the targeted therapy, a small molecule inhibitor in this case, that perturbs this kinase. So why do we need these sequencing-based approaches to personalize cancer therapy in general? Because tumors have a private combination of mutations. Oftentimes there are rare drivers, for example, the EML4 ALK fusion in lung cancer. Do we really want to develop hundreds or thousands of individual tests to monitor these aberrations, or can we do this in a more comprehensive fashion? Sometimes these targeted-based assays can miss new drivers. One of the early goals of this approach really is to molecularly enrich phase 1 and 2 clinical trials. That, I think, is going to be one of the low-hanging fruit of these types of approaches, is that we will have a better chance of our phase 1 trials of actually being successful if they're molecularly enriched. Another benefit, of course, is in the area of characterizing mechanisms of resistance, because we have permission to not only biopsy patients before they get onto a targeted therapy, but also once they progress. So you can begin to compare the mutational landscape of potential acquired mutations or acquired resistance that individual patients might have. This is also, these comprehensive approaches are also an opportunity for new discoveries, because you're having such a comprehensive assessment of genomic alterations. So this is the workflow that we've established at the University of Michigan, and this is generally available for patients with advanced cancer, those that have failed conventional treatments but are eligible for a phase 1 or 2 clinical trial. And it's both hematologic malignancies as well as common solid tumors. And this was approved again we enrolled our first patients in April of 2011. And generally what happens is that the patient meets with a genetic counselor. We obtain informed consent. We take a biopsy of their tumor, generally a CT-guided biopsy of their tumor, as well as a sample of their normal genome, which is generally a buccal swab or a blood sample. We compare the somatic alterations that occur in the tumor versus their normal genome by an integrative sequencing approach, which I'll go into in a subsequent slide. This data then is computationally analyzed through a variety of computational pipelines and then presented to a precision medicine tumor board that then deliberates upon the findings, what findings are potentially actionable in nature, what findings are incidental, are there potential clinical trials that the patient might be eligible for, or off-label use of approved drugs. We then have permission to convey these results back to the treating oncologist as well as to the patient through the help of genetic counselors as part of this general process. And again, this is not a therapeutic trial. We can't dictate therapies to any individual patients, and it's not linked directly to any clinical trials. Each of the patients will need to achieve the standard eligibility criteria for individual clinical trials based on this approach. And if off-label use of drugs are considered, we generally go through the process of running a CLIA approved test. However, we are moving our next generation sequencing-based platforms into a CLIA environment such that we can begin to report these results in general. So the basic idea is based on a mutation of an individual tumor that we will be able to match that to a targeted therapy. For example, a PI3 kinase mutation. These patients could be suggested to a PI3 kinase inhibitor trial, and so forth. A number of other interesting things that can essentially be assessed in the context of a comprehensive approach like this. So what are we doing in terms of integrative sequencing? Our basic idea here is to be comprehensive about the types of mutations that can drive cancer and that are a cause of cancer. So we try to be comprehensive about structural rearrangements, gene fusions and translocations, inversions, copy number alterations, amplifications and deletions, point mutations and indels, as well as gene expression alterations, outlier genes, different pathways or networks that might be perturbed. That's our general sort of philosophy, is to try to be comprehensive about the types of nucleic acid alterations that can occur. And this is the types of technology that we need to carry out in order to achieve this. Generally, low-pass genome sequencing of about 15x coverage, whole exome sequencing of the tumor. We're now doing 250x coverage of the tumor, matched with about 100x coverage of the normal sample, and then transcriptome sequencing. So we're carrying out sequencing of the RNA. So this combination of integrative sequencing approaches allows you to be comprehensive about the types of alterations that occur in cancer. So we're doing both DNA as well as RNA sequencing. I would say that for the first 20 patients, we actually carried out this entire process. But what we found out through the general process was that most of our mutation calls were being made in terms of the exome as well as transcriptome, such that we only carried out low-pass genome sequencing on an as-needed basis when the previous technologies did not lead to a particular result. The other important aspect of these approaches is, can we carry out these results in a clinically relevant time frame? I think it's great that you can do these approaches, but if it takes 6 months to a year to actually complete the sequencing and analysis, that generally becomes useless to the individual patient. So in the first 20 patients, we were able to actually turn these results around within a 4-week turnaround period from time of tumor biopsy, pathology assessment, sample prep, sequencing analysis, all the way up to generation of a molecular report or a molecular assessment of the individual patient's tumor. Now, since we've gone on to do about 200 patients, I would say that our average turnaround time has been more like 5 to 6 weeks. There have been a number of bottlenecks in the process, but we feel that in the future, this type of turnaround will be improved, probably on the order of 2 to 3 weeks, as the sequencing infrastructure improves or the sequencing approaches improve, as well as the computational approaches improve. The other aspect that we've incorporated in our general process is this idea of a precision medicine tumor board. We previously called this a sequencing tumor board, but we felt that that didn't necessarily have enough vision, and that I think in the future we're going to begin to incorporate other omic alterations like metabolomic alterations or proteomic alterations. So we sort of broadened our definition of what these tumor boards might be faced with in the future. But essentially they're basically the evolution of a conventional tumor board idea, but incorporating expertise in genomics and informatics, clinical genetics, bioethics, as well as patient advocacy. And generally, the tumor board meets and deliberates upon the various findings of an individual patient and considers what clinical trials might be available, what off-label opportunities might be present for that individual patient, and generally classifies the types of mutations that an individual patient might have as a class 1 mutation where approved agents might be available, say FDA approved versus off-label use. Class 2 might be an investigational agent, might be potential, either a late-stage clinical trial or an early-stage clinical trial. Generally, class 3 mutations are considered of preclinical interest, but for example, there might not be a compound that's in early-stage clinical development. And then finally, class 4 has been of unknown significance. So now just to end with a few case examples of patients that we've sequenced through our general approach. The first example is actually a discovery that we were able to actually make from this approach. And this is just one example of a discovery where we've actually discovered the pathognomonic gene fusion or rearrangement that accounts for all of this particularly rare tumor type called solitary fibrous tumor, or hemangioparasitoma as it was previously called. This was a 44-year-old woman that had surgery and radiation for an anaplastic meningeal malignant solitary fibrous tumor, or SFT. We carried out a CT-guided biopsy of this individual's tumor, and the histology was consistent with a solitary fibrous tumor. We carried out this integrative sequencing approach. Somatic point mutations were generally silent and not necessarily genes that were linked to cancer necessarily. The copy number landscape, these are all 23 chromosomes lined up here in different colors, was relatively silent. But by RNA sequencing, we identified the NAB2 STAT6 fusion with considerable read evidence that this potentially would be a contributing lesion in this particular case. An open reading frame was generated as well. And we went on to extend this by collecting over 50 cases from Memorial Sloan Kettering under Christina Antonescu, as well as at the University of Michigan. And we found, through a combination of RNA sequencing as well as RT-PCR-based technologies, that 100% of the cases had a variation of a NAB2 STAT6 fusion. And we went on to carry out a number of experiments to show in cell line models or preclinical models that these fusions actually induce a proliferative response. I won't go into the details as to the mechanistic studies that we followed up on, but we showed that this recurrent gene fusion, which is found in 100% of solitary fibrous tumors, is actually the biological cause of the disease. And basically what happens is that a transcriptional repressor gets converted into a transcriptional activator by inheriting an activation domain from STAT6. So you generate a NAB2 STAT6 fusion, which basically drives this growth proliferative disease. The second example, and in that case, that was an identification of a fusion, but of course there aren't drugs against that particular fusion. So what we're trying to do is pursue downstream things that are activated by this particular fusion, and it appears that the FGFR pathway may actually be activated directly by this fusion, and we're considering trials in that context for solitary fibrous tumors. The second case is where we've actually identified an actionable mutation in a pediatric leukemia and potentially actually impacted the life of this individual patient. This patient was a 9-year-old girl that was diagnosed with pre-BALL in 2008, failed a number of combination chemotherapeutic regimens from COG, went on a Make-A-Wish trip, and eventually relapsed again after these combination chemotherapeutic regimens. She developed a left-sided chest pain, and a pleural effusion, and presented to our program where this is the pleural effusion that we tapped, and we carried out the integrative sequencing, and we found a number of cancer-related mutations in this patient, including somatic mutations in RUNX-X1, indels in Notch2, as well as a homozygous deletion in CDKN2A. But what we focused on was the most actionable mutation in this particular patient, in that we identified an ETV6 Abl kinase fusion, which is essentially similar to the BCR-Abl fusion found in chronic myeloid leukemia. We went on to actually confirm this finding in our CLIA cytogenetics laboratory and showed that indeed ETV6 and Abl are fused, so we were able to actually report this in the individual patient's medical record. As I had alluded to, this is a relatively rare fusion, probably less than 1%, so some case reports have identified this in individual patients, but generally this is not actually screened for in typical standard approaches. So this wouldn't have actually been identified unless you took a more comprehensive sequencing-based approach. The idea here, of course, is that the Abl kinase is driving this particular disease, and that small molecule inhibitors against this Abl kinase might impact this patient. The other thing that we were able to do, since this was a hematologic malignancy, was able to actually culture this patient's cells in vitro and see if indeed the patient's tumor cells or leukemia cells were sensitive to the small molecule inhibitor imatinib. This is our positive control. This is K562 cells, which are BCR-ABL positive. They're from CML, quite sensitive to imatinib against, again, the Abl kinase. Similarly, this is a negative control here, which is resistant. When we isolate out the leukemic cells from this patient that has this particular, the ETV6 Abl kinase, they're quite sensitive to imatinib in vitro, while the same patient's CD19 negative cells are quite resistant to the disease. And we were actually able to enroll this patient in an imatinib combination chemotherapy trial, and currently this patient has been in remission for over a year based on having this imatinib treatment. And then the last example, and I'll, again, this is very abbreviated, of potentially targetable gene fusions that we've identified in 4 patients out of the first 100 patients that we enrolled in the MyOncoSeq program. We actually identified targetable fusions of targetable FGFR gene fusions in a diverse array of cancers. This includes 2 patients with metastatic cholangiocarcinoma, 1 patient with metastatic breast cancer, and 1 patient with metastatic prostate cancer. We went on to take advantage of the public domains data as part of the Tumor Genome Atlas sequencing effort in general, and found that basically this fusion appears to be present across a diverse array of cancers, which are listed here, and they all have the kinase domain spared. And what's intriguing about this is that it suggests that this subset of patients might be amenable to a small molecule inhibitor against FGFR, the FGFR pathway, because they lead to aberrant activation of the kinase. And I'm not showing you the preclinical data showing that indeed these fusions activate the FGFR pathway and are also susceptible to the FGFR inhibitor. We were able to actually refer a couple of our patients to FGFR inhibitor investigational trials. We're still waiting in terms of the potential impact on those individual patients. But in parallel, the group at TGen in Arizona, who has a similar clinical sequencing effort as ourselves, actually presented this in a session before my session at the 2013 AACR annual meeting, where they also discovered FGFR2 fusions in metastatic cholangiocarcinoma. And in 3 of their patients, they were actually able to, in an off-label setting, treat these patients with the small molecule inhibitor called ponatinib, which was originally actually developed for BCR-ABL, but was shown to have off-target effects on the FGFR pathway, very potent off-target effects on the FGFR pathway. These patients, and this is the tumor of this patient with metastatic cholangiocarcinoma, and then subsequently treated with the drug sunitinib, or ponatinib off-label. That in general, the 3 out of 3 patients had a remarkable, at least initial remission of their FGFR2-driven tumor. And this is really work done by John Carpton at TGen in collaboration with colleagues at Mayo Clinic. We have gone on to extend these general clinical sequencing efforts in a multi-institutional fashion across a consortium across the US as well as in the UK, where as part of the Stand Up to Cancer effort, we're beginning to sequence 500 patients with castrate-resistant prostate cancer, and we will basically be able to assess whether these types of clinical sequencing efforts can be carried out in a multi-institutional setting, because this will be important in the context of the development of clinical trials in the future for sure. This is just to say that this is certainly a team effort at the University of Michigan, and we have a number of collaborators across the country. And then finally, I'd like to just leave you with some challenges moving forward, and I think will sort of be food for thought in terms of the various panels that Stan has established here. And certainly our pilot study suggests these types of questions in general. What type of molecular aberrations should be monitored? Should we look at point mutations only, or do we need to cover amplifications, deletions, and gene fusions and others? What type of sequencing should be employed in terms of, should we look at RNA and DNA sequencing? Should we consider epigenetic alterations and others? Should we look at targeted panels versus comprehensive approaches? How do we handle broad sequencing in the context of regulatory approval? The previous paradigm was that you had a particular mutation or, say, gene fusion, and that then matched with a targeted therapy. For example, the EML4 ALK fusion run by a particular FISH assay matched to crizotinib. But I think moving forward, that's not going to be particularly visionary, in that a lot of these comprehensive approaches allow you to map these alterations in a much more comprehensive fashion. But then how do we deal with regulatory approval as we move forward? Another major challenge that we've faced actually with our patients is how do we end up paying for drugs used off-label when we have a compelling molecular rationale for an individual patient, where we see an FGFR fusion, but there might be an off-label use. How would we pay for, say, ponatinib use, which I think costs over $120,000 per year for that particular drug? How would we actually get that reimbursed for an individual patient? How should future clinical trials be designed in sort of incorporating that many of these mutations occur over diverse cancer types? For example, the FGFR fusions occur across a host of different cancer types. The ABL fusions occur in CML as well as in ALL. So how do you then design clinical trials to really have that as be a part of that things should be lineage independent or histology independent as we move forward? And then I think the larger sort of vision as we move forward is how do we match combinations of mutations or pathways that we see in an individual patient's tumor. So the individual patient's combination of mutations end up being quite private. How do we match that with multiple targeted therapies? I think that would be sort of the ultimate vision for the future, is how do we get to that point where you have a multiple sets of mutations that suggest a combination treatment based on having multiple pathways being aberrant at the same time, so that you're also anticipating resistance mechanisms that might come to play. So I think with that, I was just leaving you with some questions that I think will be better sort of brought out in the panel discussions, but if there's time, I might be able to take questions, or if so, we can move on, or whatever Stan would like to do.
First of all, thank you. I think you've set us up, and what I'd like to do is get the panel to start addressing these. And Arul will certainly be here, and he's not on one of the panels. We're going to make sure you get a chance to raise your hand.
Sure, I'll be right there in the front row there.
For that wonderful kickoff to the forum. This is the first panel, and we're going to be discussing how to validate genomic-based decision-making in cancer. I'm Sandy Markowitz. I'm the head of the Cancer Genetics Program at the Case Comprehensive Cancer Center and the PI of the GI Spore here at Case. We're going to begin just by allowing the members of the panel to quickly introduce themselves. Michael, do you want to begin?
I'm Michael Tainsky. I'm a professor in the Department of Oncology at the Wayne State Medical School and a member of the Karmanos Cancer Institute for the last 15 years or so. My interest is cancer genetics, particularly early detection, both genomics and proteomics, and what we call epitomics, which is a process we've developed to use autoantigens as a screening test for who's carrying a cancer and who not before there are symptoms. And because the host immune system will develop antibodies, much like a paraneoplastic syndrome, to the cancer cells' proteins.
My name is Roy Jensen. I'm the director of the Cancer Center at the University of Kansas, and my background is primarily in breast cancer research, specifically focused on BRCA1, and my clinical training is as a anatomic pathologist.
My name is Carl Morrison. I'm the director of molecular pathology at Roswell Park, and more recently the director of our newly formed Center for Personalized Medicine, where we're trying to put together many of the things that they're doing at the University of Michigan. Being in the state of New York, we meet many very significant regulatory hurdles that perhaps you wouldn't encounter at other sites. And my background is very much centered in that. I've for many years sat on the College of American Pathologists Molecular Oncology Committee that established all the proficiency standards for molecular testing. So it has a big impact on how we design tests in the state of New York.
And I'm Sandy Markowitz again. I'm a medical oncologist by training, and our laboratory works on colon cancer genetics.
Robert Penny, I'm the CEO of Paradigm, a University of Michigan company. I've been involved with molecular pathways and introducing gene expression into patient care and characterizing patient tumors, and then getting heavily involved with the Cancer Genome Atlas project to go out and characterize 20 cancers with 10,000 analyses and put them on the web to really accelerate this entire process. Very much looking forward to today's discussion.
Okay. Welcome to our panel members. Thanks for coming in. So let me begin. We have framed 4 questions for discussion by the panel this morning. And the first one is right at the beginning, what should the test be? Are we talking about doing whole exome, whole genome, RNA-seq? What exactly is the test that we are proposing that folks should be getting under the rubric of personalized medicine? Michael, do you want to begin?
It seems to me that we ought to be doing whole genome or whole exome seem to be sufficient, as Arul mentioned. It seems that's the most efficient approach to the question, but reporting out those where we have actionable agents, we should, and then databasing the rest, because with that databasing we'll find much more information among the private mutations that will seem private initially and eventually come into the few % range. And then we'll know that those are areas where actionable agents will be needed.
I suppose my response always to about what is the test would be starts with, well, how are we going to approve the test? Are we going to approve the test at the state level, at the national level? Is this going to be a laboratory-developed test, an in vitro diagnostic controlled by FDA? And until we get some type of national guidance, it's about how we're going to approve the test. To me, it's a little bit difficult to define what is the test.
I think we're going to pick that up in a moment.
Yes.
Yeah, I would say that the answer to that question depends on what you're doing the test for. And I think certainly at the discovery level and the preclinical level, you have to apply as broad a range of tests as possible, and certainly whole genome sequencing needs to be included in that. I think as you move forward in the therapeutic development process, I suspect, and I think there's lots of folks in the audience that can confirm, is that whole genome sequencing as an entry point for clinical trials is probably a pretty unmanageable beast, at least at the current time. And so, for registration trials and things like that, I think there's the preclinical biology is going to define what the agent is targeting. And I think, at least for the purposes of the trials, you should probably develop a more defined test. But then the other aspect of that is that you should certainly have material available and do genome sequencing for patients that are being put on a trial, and hopefully for sort of post-trial analysis, because as Arul's already pointed out, there's a lot of ways to skin a cat in terms of inactivating tumor suppressor genes or activating different genes. And so when you get to a point where you have a subset of patients that you think should respond or not respond and do respond, then you have some material to go back and sort of interrogate that and see why the outliers are outliers.
Bob, I'm wondering if you don't have a different point of view based on what Arul just said.
It's a complex question, and I think at the end of it all, we would love to be able to provide research findings for discovery, and that's a critical part of this. But there's also the component of not missing a patient, and when you have a positive, making sure that the patient truly is positive, i.e. sensitivity, specificity, being agreed upon with some standards as we go forward, so that we can get the right drug to the patient and help advance in a robust fashion the clinical trials piece. At Michigan, we have a rules enterprise, which is remarkable, as you all saw, and then we have a more robust targeted version that has very, very deep coverage, very rapid turnaround time for clinical decision making. And I think they clearly both have their attributes. And some people have combined sensitive tests with specific tests in a fashion to try to arrive at a means of doing this. The challenge is doing that financially. The challenge is doing that in a timely fashion for the patient and doing it with enough sensitivity and specificity that we aren't missing patients. I think the piece that always concerns us most is that we need to have some agreed upon guidelines that we come to so that we do have quality in what we're doing and we don't inadvertently miss a patient without the know-how of the sensitivity, or even worse so, introduce a drug to a patient when it's a false positive. And I think for those reasons, there are a variety of ways to get there with a series of tests or providing one test with high sensitivity, high specificity, and we try to actually look at both.
So I'll take the moderator's prerogative to have the last word on this topic point. It seems to me we had a little bit of an apples and oranges discussion going around the panel, and that we've talked a little bit about different models. I think that what Roy was talking about was you've got a drug, how do you find the patients who that drug could target? But I think that the context that perhaps many of us are thinking about is you've got a patient, and that patient has failed standard therapy and now wants to know what you can do for them. And so now you're looking, is there anything in that patient's tumor that could be potentially targeted by anything any of us can lay our hands on? And I think in that context, we're probably talking about how do you cast a very broad net. And I thought Arul's data was some of the first I've seen to actually compare the value of doing different types of omics, if you will. And I think given that we're interested in anything that could activate a gene for targeting, and that could be a mutation, that can be a structural rearrangement, that can be a change in gene copy number, that we need to be able to capture exomic mutations and things that would change expression or structure, as in RNA-seq or in whole genome sequencing. So I actually think the test has to be all three of those, perhaps stacked up in the way that Arul has suggested, where you begin with the first two and only add the whole genome in if you didn't find something actionable on the first two. So let me use that as an opportunity to segue to the second discussion point, which is this technology ready for prime time? Is this testing technically reliable? Would any of us be comfortable putting a potential toxic agent into a patient's vein based on the results coming off an Illumina sequencer? Or do we have to have Sanger confirmation before we do that? Do we have to somehow make this testing CLIA compliant or FDA compliant? Can you do that in individual hospital clinical pathology laboratories, or is this the kind of thing that can only be done in centralized reference laboratories? I guess we'll start. We went right to left this time. We'll do Hebrew and go left to right.
Sure. So, and I agree on the holistic approach that you brought up, Sandy. It's we find all different avenues are important, and you're never quite sure. And sometimes they validate the initial findings as well as you see those. So I do believe that it should be CLIA and CAP compliant, and that it's important that the CAP guidelines came out last August, and it's very good for a process. Unfortunately, they didn't address specificity and sensitivity. And I think that some guidelines and assistance to either the FDA or the CAP would be very useful for that. I think the Sanger sequencing for confirming is very good. qPCR can also be utilized for that purpose. But again, we want to be certain that the positives that we find do match up and that we've got that. Again, I do worry about missing with the depth of coverage in the initial screen. And we were trying to take a look at what would be a reasonable number there, and we threw out for discussion purposes 1,000x depth of coverage to try to get the sensitivity to be high enough that we felt that we really had a good screening test with whether you were doing whole genome sequencing or targeted sequencing.
I think the question really comes down to is how much do you believe your data, right? And the question becomes then, if you have any doubt at all about your data, should you use a second method of confirming your data before you report it, or should you use a second method of confirming your data before the patient is treated for a specific change? And there's really no guidelines out there that are on a national basis to say how we should do this, because in all states in the United States, except for the state of Washington and the state of New York, you set up a test, use your own gold standards, write down your test results, then start testing and wait till you get a CAP inspection to see whether or not you pass. That could be 1 day, it could be 1 year, okay? In the state of New York, the state of Washington, you submit all your data to the state first before you're ever approved to do testing. I'm talking about laboratory developed testing. You know, if you're talking about an in vitro diagnostic, you have to go through the FDA. But I do think we need some clear guidance on a national level about how are we going to validate these tests. Are we going to take the traditional method of working up a test and going through a validation? Are we going to go to a newer model which states, you know, there's some group out there willing to issue a gold standard, and if you can take the challenge and pass the challenge, then your test is acceptable and goes past and can be approved for testing. I think that we will find that that is definitely the more effective and more accurate way to do this, but it's really going to take some very strong group to step up and offer those standards for testing of various laboratories across the United States.
Yes, I think the CAP has done a great job in terms of really providing the metrics for standard laboratory testing, and I think that in working with them and other pathology groups, I think there's a real chance to develop the appropriate certifications and standards. In terms of whether or not we need to have centralized testing, I think in an ideal world that would be nice. But I can tell you from practical experience with immunohistochemistry, that will never happen. It's nice to think about it and dream about it, but you can pretty much forget about that. So I think we're going to have to work awfully hard to develop external means of validating these laboratories, because that's going to be the reality going forward.
I agree. I think that it's going to have to be at a local level that the sequencing is done. I think that the question is really the sensitivity of the laboratory, and the laboratory will have to pass a certain sensitivity level. My concern is more at the level of an individual sample being validated, let's say by Sanger, where you don't have the depth of sequencing ability that you have with RNA, the DNA-seq. We go as much deep as you want. And so is there going to be a different sensitivity threshold for the validation than and then what is an actionable result again? When do you put something in the vein of a patient? On validation, is it have to hit a different threshold? I think the technical balancing between discovery and validation are going to be tricky.
So maybe I can try to stir up some controversy. I think many of us who have used Illumina sequencing know that it is rife with false positives. And yet, I think also all of us who have been involved in clinical trials understand that if there's already a 4 to 6 week wait to get the result back to a treating oncologist, you're pressing the envelope in what makes practice for a clinical trial. So I think we may need to have a clinical trial in which we concede that the test falls below what has been traditionally considered the standard of care for making clinical decision making. And yet, I think that trial equally is going to have to insist that we do try to validate all of the calls so that we just find out exactly in some rigorous fashion what the false positive and the false negative rate of this test really is. Otherwise, we run the risk of potentially getting negative data because of less than perfect testing and killing a promising concept before it ever gets out of the gate.
And in that context, if we look at, if you make this rule that before you treat a patient that all these tests have to be validated in a CLIA certified approved test, we will find that probably 90% of these findings we have, there will not be a CLIA certified test, right? So if you wait for the CLIA certified test to be approved, you could be months and months and months. So at CAP for years, there has been this concept that testing should move away from single analyte validation to validation of the methodology. So if we're going to change the way we're doing this whole exome approach for cancer, we need to also change the way we validate single analyte testing, or we're going to find ourselves building this great amount of infrastructure to support genome-wide screening, and then we won't have a methodology to validate it if we don't change our single analyte approach also.
So if I could speak, I think one of the pieces that I'd like to proactively suggest for this panel is maybe we can make some recommendations as to what would be some standards that would be acceptable, whether we could come up with some gold, platinum type DNA and RNA, known mutations, known abnormalities that we could circulate and start off the process of having some specifications that we could draw as a baseline to try to address some of the issues that everyone's talking about.
I think actually, I'm not a pathologist, but I believe real pathologists, in fact, circulate samples and ask labs to prove that they can validate those samples, find what's in those samples.
Yeah, there are multiple samples across the country that you can use to validate with. You know, the group Edge Bio has put forward using all the different HapMap cell lines. The problem with those, they're normal germline DNA and they're not cancer, so they create problems. But we have, like at Roswell Park, what we've used for all of our validation is we have matching samples to hundreds of samples we submitted to the Cancer Genome Atlas, and all of those have had exome sequencing at like 150x, and they've had RNA-seq done, so we know the results. So I think you must state that if you're going to, validate a test like this, you do need a unique set of gold standards before you ever begin. And many people don't have those. They're not so easy to come by.
So that segues into, I think, the next question, and I would welcome a rule chiming in on this one. And that's, what's an actionable test result? How do you know it when you see it? Is a somatic change in cancer an actionable test result only if it's already in the literature as having been proven to be oncogenic? Or is this a technology where we're going to find a lot of private mutations that are pathogenic but that aren't in the literature?
Well, I think it's useful to maybe look at the most extreme case that I'm aware of, and that's with BRCA1 and 2 sequencing. And it's literally taking decades to figure out sort of the utility of all sorts of literally hundreds of different mutations or sequence variations in that gene. And there, I think a number of, one consortium in particular has developed a pretty thoughtful approach to this by utilizing clinical data from large numbers of families, showing that specific mutations are associated with a high degree of penetrance, biological data where you confirm that a sequence variation does have significance in terms of changing the functionality of the protein product. And it's for something like BRCA1, it's extraordinarily complicated. And I think that's probably as complicated as it can possibly get. And many other genes are going to be much simpler than that. But it's not going to be easy in many cases.
I agree. I mean, I was going to make the same analogy, that we have BRCA1 testing or BRCA2 testing. We have a huge number of variants of unknown significance that give ambiguous suggestions to the patients from genetic testing and counseling. And often they're left with, certainly wouldn't have an actionable surgical interventions, prevention, and so on, based on these variants. They have to do watchful waiting. And I think we're going to end up with a parallel in the, let's just take some oncogenes. If you get, let's say, BRAF, a good drug for it, but what if you get a private mutation and you have, let's say it's a missense mutation. So you see something non-synonymous there, you have to predict whether it would change the function of the protein. And there just isn't time to do that experiment in most cases in real time. And I think there is software that allows one to predict that. And I just don't feel comfortable having it as an actionable result on a private mutation that's in an oncogene that you know is a target for drug therapy, but you've got a new variant. And I think as in the case of BRCA1/2, it's a variant of unknown function, and it's, of course, now it's a somatic change rather than germline change. But I think the questionability is still there, and I think we have to be very cautious about.
Bob is jumping out of his chair here on my left.
It's my normal state. I think it's helpful. One broach that might be deemed as of value would be to first have a rigorous informatics solution as to what has been published and what has been graded. The U.S. Preventative Task Force Services, established by Congress, appointed by Congress, and supported by HHS, has some guidelines that we're very familiar with, and putting together a very robust process that allows a real-time analysis of what our colleagues are doing in the literature as a baseline to draw from helps you then make that leap, because you're going to very quickly get into on compendia, off compendia, clinical trial. There's going to be multiple pieces of this. Geez, we can't get this patient in the trial because of these challenges, but what can we do because we have this level of evidence? And there's a practice of medicine that's involved as well. There's level 1 that everyone's comfortable with. When you get to the 2-1, the 2-2s, it gets more difficult, and it's clearly going to be dependent also on the type of cancer. If you've got a pancreatic or a high-stage, very lethal disease versus a late-stage disease, and the whole compendium that one comes across. So I think it would be of value to look at where do we start from a robust process and then explore into the variant calling and how we address those challenging issues that we all faced with. Is it in a research design? Is it not in a research design? And can we use this to build the information for tomorrow?
I wonder if I could call on Arul to comment on how often in the patients you've looked at are you finding new mutations in targetable kinases that haven't been seen before?
Yeah, so I think in our experience, we're certainly screening a variety of different cancer types. So I think things could sort of get washed out from doing so many different cancer types, and you'd have to look at the subset analyses of this. But the definition that we like to use, just so that we're more encompassing, is generally actionable or informative, because that generally captures things that are potentially involved in the biology of the tumor, but for which you don't necessarily have a drug. So many of the tumor suppressor type mutations and so forth, certainly contributing to disease progression, but for which you could not necessarily act upon with a particular drug. In some cases, there are certainly possibilities. I would say out of the first 250 patients or so that we've sequenced, that about 50 to 60% have things that would be actionable or informative in nature. Certainly there are mutations in the other 30 to 40%, but we don't understand the significance of them. They might be in tumor suppressors that we don't understand, or they might be oncogenic activating mutations and so forth. But out of the, say, 50 to 60% that we identify something that is either actionable or informative, I would say that the actual percentage that is sort of truly actionable, for which there's a mutation and likely a drug in play is probably much less, probably on the order of maybe 5 to 15%, where we can actually take an action upon the effect. And the other aspect is also sort of how do you define this? The other aspect is that we don't understand the off-target effects of these kinase inhibitors. So we're always wondering if we do have an activating mutation in a kinase like the FGFR pathway, say, is there a kinase inhibitor that's already out there and approved but then has an off-target effect that hits that particular mutation? So it really depends on how you define actionable. So that's why we like to use just actionable plus informative so that we sort of cover that aspect.
So I think I too will try it. Oh, I'm sorry, Michael.
Well, I think there's another level of heterogeneity besides the mutation location and whether it has some biological connection, is the heterogeneity in the patient. You're going to biopsy a lesion. How many biopsies are you going to do? Do they all have to match? If a person has heterogeneity in their different biopsied lesions, at the particular so-called actionable, is it still a patient you want to treat because you know it may affect one minor lesion but not really do anything for the patient? Is that appropriate to implement? Is that actionable still? I think that's a question. It's almost an ethical question.
I think that's an interesting question, even if you only biopsy one site, which is how big does the clone have to be before it's actionable? If it's a 2% variant, is that actionable? But I think that maybe I'll again comment that if one of the objects of the panel today is to persuade Dr. Durashow, who's sitting there in the front row, that he wants to invest, a lot of largesse in sponsoring a large clinical trial to do this, then I would argue we probably need 2 tracks in that trial. And one of them is the rigorous track, where we really only treat a mutation that is clearly oncogenic and for which there is a drug that we know will work. And the argument there is that will tell us how often treating BRAF is treating BRAF in melanoma and how often it's treating BRAF in colon cancer where it doesn't help anybody, right? And then there's got to be another track, which would be the compassionate use track, which is, the first time that a rule or somebody else sees an somatic EGFR mutation that no one has seen before, right? But you've got a patient, and what does he have to lose if you put him on an EGFR tyrosine kinase inhibitor? Wouldn't you do it, for your mom? I think we're going to have to be rigorous about separating those 2 tracks out, because it's the first track that's going to give us the numbers that we can trust, and it's the second track that may inform what the next study should look like.
Pat, you look like you're just pregnant with possibilities.
I would try to read this stuff, but one of the problems that you have is also when you have a hypermutated tumor. First of all, with multiple, heterogeneous tumors, typically the actionable mutations are consistent. At least reported cases have been consistent regardless of the different biopsies and the heterogeneity of the tumors. But when you have a hypermutated tumor, the question is, and you have a lot of level 1 actionable mutations, which of those do you go with? And I think one of our greatest limitations right now is combination therapies. Because the other issue that nobody's addressed yet is when you have a bunch of level 1 actionable mutations, what is the ratio of those mutations or the treatment ratios that you need to impact in order to give you the greatest benefit of response? Very complicated.
Yeah, Pat, I would, I think that situation calls for a whole different type of clinical trial than what we have in the armamentarium right now.
And yes.
And it's, and I think people do need to come together to figure out that exact, question. It's, somewhat like the I-SPY, trial where you're really trying to adapt to a very fluid situation and assigning targeted agents, based on a rules work or other folks' work in terms of sequencing and say, we're going to pick out the 4 most likely actionable items that we have targeted agents for, and we're going to treat these patients, with that and see what happens. And I think, But there's no mechanism right now that allows us to do that.
No, I mean, it's going to take a lot of ingenious trial designs. But one of the advantages that Laura has is she's doing adjuvant or neoadjuvant. And as a result, if you look at longitudinal, she's probably got the least amount of mutations of any subset of tumors that are moving forward in this clinical trial type of field right now.
And I also think that something would help us from a laboratory perspective if someone would define what are the actionable variants. Like my cancer genome at Vandy has really gone out there and done a lot of effort at this, but there's still a lot of parts of that that's not completed. And if we had some type of national initiative to say, what are the actionable variants for each particular tumor type to be tested, that would go a long way.
So
Yeah,
Max, go
hi.
ahead.
Max Wisher from the University of Michigan. I want to pick up on a point that Pat just made and ask some of the panel for their opinion about this, because we've really been talking about mainly patients who have very advanced disease that's been heavily pretreated, which is, of course, going to be the hardest one to target because it is so heterogeneous. So the question is, what do you think about the paradigm of actually much earlier testing, a matter of fact, when we're going to actually be testing patients up front so we know the mutation and can use a more logical sequence of events, of treatments, where patients may go to targeted therapies really early rather than late, potentially, eventually in the adjuvant setting, while we have much more of a chance for actually long-term benefits.
I think you also get down to the question, what's the minimum variant allelic frequency for which you will treat a particular mutation for? And who's going to make those guidelines and judgments?
I think Max, though, has raised an interesting question because it sort of upends the traditional pyramid of oncology. And I think you could make an argument that for diseases for which the expected survival is 6 months or less, why not? Right? Pancreatic cancer, esophageal adenocarcinomas, we can easily enumerate the ones for which the standard of care does not offer much of a survival benefit, or if it does, it's measured in weeks. There are other questions, but I'd like to actually move well, go ahead. Gerald, you've got, that'll be the last comment, and then we'll go to the last point.
Gerald Pepper, Karmanos Cancer Institute. Yeah, I just wanted to pick up on this question with early treatment. So my expertise is lung cancer, and I just want to throw a caution in here. So there are actually adjuvant chemotherapy trials in lung cancer that are using targeted agents for EGFR mutated lung cancers early on. And the preliminary results show that it's a completely different beast. Actually, if you give erlotinib to a patient in the adjuvant setting that has an EGFR mutation, you actually hurt the patient. I just wanted to throw this in there. It's not,
So, Sandy, if I can just throw out, jumping on what Carl brought up and Max. So if you have a stage C colon cancer and you've got a 7% mutation in KRAS, how do we build the data to find out if we're going to go the route of the EGFR story, or if we combine that with the standard of care, we have a better outcome and can really early on get into the karyotypic heterogeneity before it starts getting too far out?
And with previous Sanger sequencing, we would not even know that patient existed.
But we do now.
Yeah.
And we know the percent pretty exactly.
So again, I want to make sure we separate the apples from the oranges, because I think we all intuitively understand the patient for whom we don't have good options and why that patient should get everything sequenced and a drug for which we have no clinical data. I think it's much scarier when you start talking about a stage 3 colon cancer patient for whom you have a potentially curative standard of care and wanting to change that without the context of a randomized clinical trial with large numbers in each arm that will compare whether you're doing good or doing harm. And I think we need to separate those two types of patients very clearly in our minds, again, because the first time we do harm, we potentially kill a promising field. Right. I want to just then use that to comment to segue to what I think is the last and in many ways the most important question, which is, if we're all going to be doing this, how do we collate the data so we can all learn from it? How do we find out whether, if you're going to give drug X for mutation Y for tumor Z, whether 5 guys have already done this in Florida and gotten nowhere, or 5 guys did it at City of Hope and everybody got a CR. Shouldn't there be some expectation on the part of whomever is going to be paying the freight for this, that we do it in a way so that the community can learn the outcomes as fast as possible?
I don't think that's a question. I think it has to be databased and available nationally. Because the ends in any one institute are too small to make a generalization.
But how? There's no precedent for it.
Well, I think a big issue is who's going to pay for it, and I think what you're talking about, Sandy, is something along the lines of a post-approval registry where you have to submit tumor tissue and then look for expand it to a much larger cohort, basically. And, that's the only way you're going to be able to answer that question, I think.
And we also need to make sure that the laboratories are included in that database, because make sure that we're all calling the same mutation, that we're all using the same HG build, that we're all using the same terminology so that we can communicate. And someone has to code all of those mutations so that we can compare them.
Yeah, I would agree. The agreement on specifications are critical to it, but you've got to collaborate and pull these together, and there also can be a value piece for not only coming together on those, but also coming together on more rigorous, scientifically driven ones.
Okay, there's a sign in the back telling me that it's time to allow questions from the assemblage. So I'll do that before they press the button that's wired to the electrode on my body.
So this has been a fantastic discussion. I think we've talked quite a bit about DNA level-based actual mutations, and we know from TCGA and from a lot of our own sequencing there are a lot of patients who just do not have those. So it's important that we begin to learn how to leverage the RNA. And I think Dr. Schneid's group published a fantastic paper in Cancer Discovery a couple months ago on the use of outlier kinase expression as potential targets for precision medicine. So my question is, and maybe to Dr. Schneid and to the group, how comfortable are the sequencing tumor boards in associating drug with an outlier kinase expression independent of not having a DNA-based copy number mutation or a somatic mutation and using that drug association, which I think happens quite more often than we can expect?
So I think any of our RNA-based tests are always been plagued by the quality of specimens that we have to start with. And I think that's one thing that was never even discussed here. If we're going to do to this, large-scale integrative sequencing, we have to collect high-quality specimens for these patients. And today, what we're applying clinically mostly is targeted sequencing, because we can rely on these low-quality FFPE specimens. So I think that, in my opinion, to address the question you have asked, we first have to everyone needs to develop this very exact way of procuring high-quality specimens and putting standards around those, and even qualifying the specimen before you start doing RNA testing.
Yeah, but he's asking a really tough question, which was,
yes.
what do you do with overexpression of a kinase where you don't have gene amplification or you don't have a fusion? And how do you define overexpression? Is it is it 2x, 5x, 10x, 100x?
Yeah, I think it's that's definitely a great question. And in terms of our precision medicine tumor boards, certainly I think the initial discussion is when we do have a genetic aberration, either an amplification, point mutation, or rearrangement, or what have you. But oftentimes for an individual patient, we run out of those types of mutations to focus in on. And we then, sort of the second tier of aberrations that we look at are basically gene expression alterations, where we have outlier expression, which we don't necessarily know the molecular basis of, because oftentimes we have not carried out whole genome sequencing to see if, for example, the promoter has been switched and the RNA doesn't manifest as, say, a chimeric sequence. So I think it really depends on the context and the individual sort of patient and what the options are available for that patient. If you see outlier expression, and generally it would be log-fold expression over the normal lineage, I would say, or sort of across a compendium is what we use to define outlier expression, and how compelling is that outlier. And we haven't sort of defined precise criteria because it's often subjective in nature, and that's why we do carry out these discussions as part of a precision medicine tumor board, because oftentimes these decisions are relatively gray in nature. But we were looking for sort of outlier log-fold expression changes of, say, a kinase, which would then suggest potential sensitivity to, say, a kinase inhibitor. But I think that's certainly one aspect that needs to be considered.
I think one answer is we don't know the answer, and that therefore any clinical trial that is designed had better include this as a specific outcome that's going to be interrogated.
Yeah, we used and published in JCO a prospective clinical trial. It was small, but when we looked at the expression, much like we're now doing with the next-gen sequencing in the RNA, we compared it to the normal, and we looked for a P level less than 0.01 as it compared to the normal, and used that to try to select out patients. And we had some good progression-free survival results that showed from that.
Just a general comment about, I think, the need for sort of a national database or repository. I think that's an important effort, primarily because, again, as was indicated, the ends are quite small at an individual institution. But the fact is that one important aspect to suggest that something is a driver oncogene is to see recurrence of that particular mutation, the same, say, amino acid change at the same region of the molecule. That would often give you evidence, if you even see it in 2 patients, that that was indeed an oncogenic driver because of evolutionary selection to actually get that particular aberration. What we see in tumor suppressors in general is that you'll see the mutations peppered across the gene as being inactivating and so forth. But in general, with these activating oncogenic mutations, they'll either be recurrent in nature at sort of the similar residues, conserved residues, or oftentimes create a functional equivalent or a kinase equivalent that might have diversity in the types of 5' partners, but in the end create a recurrent kinase fusion. But I think that these national registries will really help in sort of maybe even making discoveries of recurrent driver mutations as well.
Let me just remark, I think this is one of the reasons that we really do need some sort of clinical trial context for this effort, because the ability to do what the questioner just asked has been around for several decades, right? I mean, we've all been running microarrays for a long time, and we could have at any time during the last 20 years have said, let's use a microarray result to pick a drug for treating a tumor. And we didn't, because most of us in the lab know that 9 times out of 10, if your microarray tells you something is turned on in a cell line and you knock it down, it doesn't have any effect, right? So it's very sexy in the context of next-gen technology to talk about treating outlier kinases, and I think it's an appropriate question for a clinical trial. I think also it's important, though, to define it as the question so that you don't inadvertently lump it in with a different question of treating a somatic mutation, and again, amalgamate 2 types of data, one which may work well and the other one which may not work well. And you don't want to be doing a subset analysis after the study to say, well, if our criteria for treatment had been this, we would have gotten much better sensitivity and specificity.
Sandy, thank you for those comments, and your electrode just went off, so we're going to move on.
I'm suitably stimulated.
Move directly to the next panel. Rick Chilsky will be leading the discussion. And I want to appreciate the group, and we'll have a round of applause while we transition quickly. So let me introduce the next panel as we get it up onto the screen. This is the challenges for the FDA and CMS drug approval and reimbursement for off-label drug use. We have some logistics that we're going to be taking care of this morning, and that is that given sequestration, which we're all living with, we have a number of our federal employees who are video conferencing in, and I'm not exactly sure how this works, but hopefully we will hear from our off-site folks imminently. One by cell phone and one by, I hope, video conferencing. So I'll let Rick introduce the topic and his folks on the panel discussion.
Okay, great. Thanks, Stan.
I just want to take a moment to really thank Stan for having the sort of the foresight and the energy to put this this whole forum together. I think the topics here are incredibly important to all of us, and the discussion sounds like it's been really outstanding so far, even though I came in a little bit late. So we're going to segue now into discussion of some of the more regulatory and reimbursement issues relating to both how a test can actually be performed in the clinical setting and paid for, and if an actionable result turns up, how does a clinician actually get hold of a drug that might be useful to treat their patient? So I had sent some slides with, I don't know if we have them, with the panel introductions, but anyway, if we don't have them, we have obviously two people here who are quite well qualified to introduce themselves. So Neil, why don't you go ahead?
Thanks, Rich. Neil Maripol. I'm a medical oncologist at University Hospital Seidman Cancer Center and Case Comprehensive Cancer Center. I'm a clinical trialist, but I also study how patients make decisions about their cancer treatment, in particular about whether to take part in clinical trials.
My name is Jim Dorcio. I'm the Deputy Director of the NCI for Clinical and Translational Research, and I'm a medical oncologist, and I still go to clinic half a day a week.
And for anyone who doesn't know me, I'm Rich Shilsky, and I no longer go to clinic half day a week since becoming the Chief Medical Officer of ASCO a few months ago, and before that had a long career at the University of Chicago and spent a lot of time doing clinical trials. And now I believe on the phone we have Liz Mansfield from FDA. Liz, are you there? Okay, and what about Tony Murgo from FDA? The sequester is worse than we thought. Not only can't they fly, they can't even make a phone call. And what about Jeff Roche from CMS? Oh, there they are. Oh, there they are. Oh, there he is. There's Tony and Liz. Can you guys hear us? Well, we can't hear you. So we have a little technical problem there.
Something yes.
And do we have Dr. Roche? He was also to join us from CMS, and we were also going to have Martine George from Pfizer, who unfortunately had a death in her family, and we were informed just the end of last week that she wouldn't be able to attend. All right, so we are hopeful of resolving the technical problems.
Hi, Dr. Shilsky, this is Jeff Roach.
Oh, great. Good morning. Thank you for joining us. So we had prepared a number of questions that are in your materials that we thought we could talk about in no particular order, but they relate to a number of important topics. One is the topic of development of companion diagnostic tests, and two issues there that seem to be of some concern in the community. One is, if you test your patient's tumor using an alternative test to the one that is actually listed in the drug label, is that technically then off-label use of the drug, because the drug is actually only labeled to be used with the particular test? And is there some risk then that reimbursement may not follow through? The second and perhaps more important question is, how do we get innovation in test development? So if you have a test for a BRAF V600E, say, that is in the drug label for vemurafenib, as it is, and a test developer wants to try to introduce a new and improved version and get their new and improved version into the vemurafenib drug label, what is the process to enable that to happen? So that's a question we'd like to see addressed. We put out there for discussion this whole topic of N of 1 observations. So, we've been talking about that already this morning. As we make observations on patients who have their tumors profiled,
have an actionable result, have an action taken, and that produces a beneficial outcome for the patient. Is there ever a point at which enough of those anecdotes can be accumulated if based on a single test or a group of analytically validated tests where an actual drug approval might be considered as a result of a sufficiently robust response rate? We wanted to talk a little bit about patient access to drugs. And I think this is where the last panel was just going at their conclusion. There are various ways in which people can get drugs, and most of them are going to be in the off-label category, for this type of molecular profiling. So when the patient's tumor is profiled and an actionable result is suggested that recommends a course of action, generally speaking, we can expect that the drug that would be recommended would not be used on label, therefore might have to be used off-label if it's even a commercially available drug, or patient might have to be referred into a clinical trial of such a drug, or a company that's making such a drug might offer it up for a compassionate use program. But there are many issues surrounding how we make that happen, how it gets paid for, who pays for it, how does the information that results from the use of the drug in those circumstances, how is that information captured, as the last panel was discussing. So I don't have to read all the questions to you because they're all in your folder, but I wanted to just sort of set the tone for what we hope to discuss during this session. And I thought we could start off by asking each of the panel members to make some brief comments, really focused in whichever of these areas they feel most comfortable. And then we would have some discussion and open it up to the audience. So Neil, do you want to start off?
So you set the stage for a day-long discussion.
Yeah, you have 3 minutes.
Okay, thank
So
you.
pick a topic and go for it.
So I guess I'll start with levels of evidence and approval and payment. And I think it's important that we recognize that drug approval in the United States for marketing is distinct from how we pay for those drugs, and we have to think of them both differently. In the United States, and obviously we have people on the phone who might be more expert in this, but drug approval is based on benefit and risk. Drug payment, at least at the level of CMS, is based on reasonable and necessary. So, I think we've alluded to in the discussion already this morning the fact that some of our regulatory frameworks may need to change in terms of how we move forward in this era. With regard to drug approval, it may be that we're going to need different tiers of drug approvals, so that if a drug is not already marketed in the United States, but it's a drug that seems like it might have utility based on a pathway analysis or a modest amount of data, I think we do need to start thinking about what regulatory path, and perhaps it's a new definition of approvability based on these N of 1 experiences. So could a small aggregation of clinical experience in a well-defined scenario, which is very heterogeneous by its nature because you're talking about different types of cancer, could that lead to some type of approval, meaning that the drug becomes available for use? The payment side is completely, it's related, but it's distinct. On the payment side, the question is, who are the stakeholders who would stand to benefit from paying, and then how should it be set up such that they pay? And there are a bunch of different options. I mean, one stakeholder is the government, because the healthier our society is, the better. And you can imagine a scenario where there's payment with the provision that you're going to collect data and contribute it to a repository. The producers of drugs have a vested interest, because from the standpoint of pharma and biotech, the whole financial model of drug development in cancer is being thrown on its ear, because you no longer have the potential to treat 200,000 patients with lung cancer, all with the same drug in first-line therapy. And so the entire model for drug development is going to be altered. And so there's a vested interest in finding a way in developing evidence that drugs being developed by the developers are of benefit. And again, they might pay with evidence collection. And then you have, so you've got the payers, you've got the producers, you've got the government, and then there are commercial payers. And I can imagine a scenario where large insurers who are now being bombarded with requests to pay for genomic tests and subsequently for drugs to treat patients might stand to benefit by entering into an agreement with, say, cancer centers or other groups that take care of patients to pay for particular drugs in a well-defined evidence-collecting scenario. And I guess those would be my main comments.
I think that's a great way to start off. I think the way to perhaps focus the discussion in this panel is to really talk about the evidence base that is necessary for regulatory agencies to make the decisions that they are required to make. FDA has to make decisions about whether or not drugs should be introduced into the market. CMS has to make decisions about whether treatment should be paid for. In both cases, I think we would all agree that we want those decisions to be as strongly evidence-based as possible, because we all benefit as a society when the decisions can be as well informed as possible. And then the question becomes, what's the optimal way of obtaining the necessary evidence? Because I think it also is becoming very clear that we're no longer going to be able to learn everything that we need to know from doing prospective clinical trials. We're doing too much molecular subsetting, so in a sense we're creating too many different diseases that don't have enough patients represented in those disease categories. We have too many drugs, we have too many tests, we don't have enough time, and we don't have enough money to study everything that needs to be studied in a clinical trial. So we need some alternative mechanisms to gather information, and then we have to discuss the validity of that information and whether it is useful for informing regulatory decisions. So having stated that, maybe it would be good to hear first from our colleagues at FDA and then our colleagues at CMS. I know you guys can hear us. I'm still not sure if we can hear you, so let's give it a try.
Can you hear us?
Yes. All right. Great.
Okay. Yeah, we're here.
So, Tony or Liz, do you want to start off a little bit about some of these issues in terms of the evidence necessary to inform the decision-making that FDA has to do around drugs and tests, and are there alternative ways of developing that evidence other than prospective clinical trials?
Well, I guess I can start off. Liz Mansfield, I'm the director of the personalized medicine staff in the Center for Devices. What we typically are looking for is sufficient evidence that the test adequately informs the use of the drug. Test side, does that have to come from a clinical trial? I'm not talking about approving the drug, just the test. That's the way we've done it. I can envision it being done in a different way. I think we've talked with ASCO and others about creation of a database of molecular changes and patient responses to different drugs and so on. I can't say what the threshold would be of enough. I think beauty is sometimes in the eye of the beholder. So what's enough for me might not be enough for you, or the other way around. So, I think there's potential for other ways of gathering evidence. I'm not sure, where people would draw the line, though, on how much evidence was enough.
Liz, before we turn to Tony, can you offer any specific comments on this issue of how to introduce a new test into a drug label as a companion diagnostic when there already is an existing label test?
Yeah, so in the case of HER2 and Herceptin, there are a number of tests that are approved now to do that. And so it's possible to do. One of the things that you had in your introduction.
Uh-oh, we lost you. We're not hearing you anymore.
You hear us now?
Yes, all right, now we got you.
Don't know what happened there, but I'll move a little closer. In terms of saying...
Sorry, we lost your audio again.
Hello?
All right, now we've got you back. We're going to spend our
I don't know
panel
what the
time
problem
doing
is.
this.
Yeah. Can you hear me now?
Yeah, go ahead.
So maybe it's just what I'm saying that it doesn't like. So one of the questions is, if you want to say that you have a better test, I want to know from the FDA point of view, what does better mean? Does that mean you can get a better analytical measurement, a more accurate analytical measurement? Does it mean that you can test more analytes? If you had 5 mutations, now can you test 10? Does it mean that the test is more sensitive, that you could now detect 1% mutation in a background of non-mutated, whereas before it was only 5%? So those are important questions. And then how is that improvement measured? How do I know that it's better? If you want to say that it's analytically more accurate, how are you going to show me that? You probably need a gold standard or some kind of comparator. If you want to add more analytes, I think realistically you need evidence from patient outcomes that those additional analytes actually mean something in terms of therapy. And higher sensitivity, we've seen a lot of claims about this. Oh, our test is more sensitive. We actually don't know what that means because patient outcomes are unknown. Is 5% enough? Is 15% enough? Is 20% enough? Is 1% enough? We don't know because nobody's ever actually tested that. So if you want to say that a test is better, you have to define, in a sense, how it's better, and then tell us how you're going to give us the evidence that it's better. If you want to bring something on the market that's essentially the same as something already there, what we did for HER2 was look essentially at correlation studies. How well do the tests correlate with each other when tested on the same sample, which is not the greatest way to do things, because if you get discordance, you don't know which test was wrong.
Great, that's very helpful. Thanks so much. So Tony, comments from you on the drug side?
Okay. Well, this is Tony Margot.
We're only seeing your right shoulder, but at least we can hear you.
Okay, I didn't realize you could see me at all. I think you would rather look at Liz than look at me. But anyway, so I'm with the Center for Drug Evaluation and Research, so we deal with the drug end of things. And I also work a half day in clinic thanks to Jim Dershow that continues to allow me to do that. Let me speak a little bit about the question about the N of 1 studies, and specifically the question that was put forth about having evidence from a series of N01 observations, and can that lead to support for drug approval. So I'm assuming that this means when the question about drug approval applies to both supplemental indications for a drug that's already on the market, and also for supported approval of a previously unmarketed drug, a new molecular entity. So I'll try to address both of those. So as you know, by law, the FDA can approve a drug if there's demonstration of safety and effectiveness from adequate and well-controlled clinical trials. Now, assuming that we're talking about a drug that's already on the market, and assuming there's already adequate safety data or safety information that could be applied to a new indication, the remaining question then is whether data from a series of N-of-1 studies or N-of-1 observations are adequate and well controlled to demonstrate effectiveness. So in the absence of randomized control trial data, FDA has considered non-randomized trials to be well controlled, provided that there is an adequate or reliable historical control. And in oncology, demonstration of objective response can be considered historically well controlled, since with rare exceptions, tumors don't shrink on their own. The natural history of tumors is to grow, not to shrink. So therefore, response can be attributed to the study treatment, assuming that there are no other effects that that don't allow isolation of the study treatment effect. So the adequacy of data to support approval is based on a number of factors, which I won't get into all of them, but include how well the patient population is characterized. Here we're talking about if this patient is selected based on a laboratory test, it's important that the test be robust. And it also depends on the magnitude of the treatment effect. The labeled indication requires a diagnostic test. Approval of the drug is likely to be contingent on the FDA approval of the test. There's some rare exceptions to that, and I won't necessarily get into that, but the important thing is, well, nothing to point out. To answer the question, N-of-1 observations are important, or may be important elements of a drug's approval. Now, if we turn to the new molecular entities, then the same approach with new molecular entities that I just described would require substantially larger studies because they're to gather the necessary safety information. So the hurdle is quite larger for new molecular entities, and quite possibly might require some kind of a randomized trial. But the bottom line is, to the question of at what point does clinical evidence derived from a series of N-of-1 observations that show high drug activity become sufficiently persuasive to support drug approval? Simple answer to that question is that when the data are sufficiently robust to demonstrate safety and effectiveness for the proposed indication. Now that's a simplified answer, but what I'm saying is that there is the possibility for these N-of-1 studies to at least support a supplemental indication. If you look at the indications that we have for imatinib, if you go through some of those niche diseases like dermatofibrosarcoma protuberans and some of the other conditions, the other hematologic small population indications such like HES or I'm blocking on a couple others, but if you look at the labeled indications, the number of patients that were able to support those supplemental indications was quite small. Now, I don't know if that answers your question about the N of 1 studies, but
yeah, Tony, I think that your answer is very helpful. I mean, you know, it's perhaps not, you know, not surprisingly, you know, I think our colleagues in the regulatory agencies are giving us the response in both cases of it depends, and that's true. It depends on, you know, the quality and consistency and robustness of the data, but what I'm hearing is that it's also possible that you know, certain types of regulatory approvals could be based upon data that are obtained from studies other than randomized or prospective clinical trials. And I think that's an important take-home. I wanted to turn to Dr. Roche at CMS, and hopefully you've been listening in, because I'm curious to get your perspective on some of the issues surrounding reimbursement. And in particular, I know from discussions I've had with a number of people at CMS that one of the things that you guys are struggling with is the rapid proliferation of molecular tests in the marketplace, either laboratory developed tests or tests being developed by various commercial firms, many of which are being offered for sale, are being ordered by physicians, where the usefulness of the data derived from the test is variable, in some cases highly useful, in other cases much less so. I know you all are being asked to make decisions about whether to pay for such tests. And perhaps you could talk a little bit about you know, how you evaluate those requests and what kind of evidence would be most useful to CMS in making those decisions. And hopefully you heard the question. Okay, so I don't know if we have Dr. Roche.
Dr. Shilsky, thank you very much for inviting me to be part of the panel today.
You're welcome. Did you hear the question I just raised?
I'm not taking long pauses because of dyspnea, but because there is a 7-second delay on the line. Let me suggest that CMS, like any other agency, when confronted with a new situation, tends to go back to what it knows.
Which is how to say no.
Now, at CMS, what we know is we tend to stay out of trouble when we obey the dictates of Congress. And specifically, in this area, Congress has instructed CMS to devise a mechanism to use what are called compendia which allows CMS to essentially take advantage of evidence that has already been gathered and collated in these various different compendia for drug use. Beyond that, CMS, as it does for other types of new tests, puts a lot of responsibility on the local Medicare administrative contractors. Now, in looking through this section, which, for those who are interested, is section 50.4.5 of Chapter 15 of the Medicare Benefits Policy Manual, states that we can also use results of articles published in peer-reviewed journals, and especially a particular list of those journals. And our hope is to find that the evidence, in whatever form we find it, supports improvement in clinical outcome for patients who are treated with that drug who have that particular cancer mutation. There is nothing in the benefit policy manual, that would rule out a specific genetic marker for response to a given drug.
Jeff, I'm going to interrupt you at this point because the transmission is so delayed. It's consuming a lot of our time.
So as a first answer to your interesting questions, my thought would be that we would tend to follow our current policy about the off-label use of FDA-approved oncologic drugs.
Okay, thanks very much. I want to give Jim Dorshow a chance to comment. Now, Jim, obviously we know you're not at a regulatory agency, but you are at a research agency, and I guess the question to you is your thoughts on how do we go about developing the necessary evidence to allow your colleagues in the regulatory agencies to make well-informed decisions?
So let me just say, I have Stan is really making me work for my honorarium that I can't accept, because I'm on several panels.
At least you got a trip to Cleveland.
Right, I did. Right. So I'm not going to talk about, I have two other chances, I think, to talk about clinical trials, which I will do and try to answer at least part of Sandy's question. But let me talk about the N of 1 issue and what we're trying to do from a research perspective. So some of you know that the NCI has been working for the better part of a year on a variety of precision medicine research activities, one of which is the so-called exceptional case program that really was stimulated by the wonderful examples that Dr. Chinayan presented, by examples from Memorial Sloan Kettering, from WashU, where remarkable results in terms of responses in individuals after the genetic information was available came due. And so the question was raised and has been, we've been studying this for quite a while, and we've made a significant amount of progress to ask the reverse question. As, again, many of you know, I also kind of oversee CTEP as part of the Division of Cancer Treatment and Diagnosis. And over the past decade, CTEP has conducted over clinical trials with over 10,000 patients accrued to targeted treatments either in the phase 1 or phase 2 setting, and even more patients have been treated in the context of phase 2 studies in the cooperative groups. And so we did, as a first pass about now 7, 8, 9 months ago, looked back 10 years to find whether or not there were exceptional responders, complete remissions, prolonged partial remissions in targeted therapeutic studies for which we had excellent clinical data that we wouldn't have to pay for again, that could potentially be linked to FFPE specimens and then evaluated in great depth to try to understand the reasons for these unexpected responses. Again, there are many in the audience I know who have, as I do, a CV full of publications that they all end with the same paragraph. It says something like this: this drug did not have sufficient activity in this disease to warrant further testing, even though the trial may well have had some complete response responders, but in almost all cases had less than 10% objective, partial, and complete responders. And the question really was, as we initially posed, whether could we find a number of such patients and such trials, and the answer is yes, that there are well over 100 such patients with very profound responses to targeted agents, all of which were conducted in NCI-supported clinical trials, many of which have never been published because it was 5 responses out of 60 patients in disease X or 4 out of whatever. And those are just for the targeted therapeutic studies. There are many more that go back into the late cytotoxic era, if we might call that, again, outstanding responses, but again, some published, some not. And so the question is how to construct an effort and how to support an effort in which not only from our cooperative groups, and our cancer centers participating in the phase 1 and 2 programs, but also across the CCOP network and elsewhere, could we collate a series of these so-called exceptional responses, and then utilize the TCGA laboratories to do very extensive evaluation of those patients, again, from whom, most of which, we have clinical trials. And so all I can tell you is that we've made, I think, a lot of progress in planning for, organizing the resources for, and developing some of the processes for going forward with such an activity. It is not being done in the context of trying to get a regulatory approval, but in fact being done to provide a national database, because all of this information will be publicly available, to spur us, and who knows? I think the answer is nobody knows just how profound and what we'll find in this context, but I think we're going forward with that. The biggest issue is not finding the money for this particular enterprise, because unfortunately there aren't 2,000 complete remissions in mesothelioma that we missed. But on the other hand, the question is, I think we will have the resources, we will find the cases for this initial type of sequencing and other kinds of analyses that will be done on those tissues. But the immediate next question will be, if I don't say this, you'll ask me, is, well, how do you validate those results? How do you then do the subsequent, none of those wonderful examples that you're all aware about simply related to the demonstration of a particular mutation in a particular gene in a particular clinical trial. They all had some beautiful, beautiful molecular biology done to actually substantiate in additional model systems, whether they're cellular, cell culture, or animal model systems, or all of the above, to really validate that this was a novel target. And that's, again, a matter of a very important discussion, one that we're actively engaged in, because we want to move forward. It would be a tragedy not to actually find subsets of patients that would respond to drugs that may even be commercially available, or if not, were for which all of the safety and a lot of efficacy data are available, that could be resuscitated if we had the subsets to investigate. And so really our view is that we need to help and support the extramural community to develop the data that would allow some of these activities, perhaps some of the drugs out there that were not brought forward commercially, to actually have a commercial basis.
Thank you, Jim. So we're going to take just 1 or 2 questions, I guess, from the audience, because we're already past our allotted time for this panel, and apologize for all the technical difficulties we had, although I had nothing to do with them, but I'll apologize anyway on behalf of the meeting organizer. So, question, yes.
Hi. Yeah, thanks very much. You touched on these issues a little bit, but I wanted to get a little more clarity if possible. One is for development of new drugs, to get approval for marketing, you have to have efficacy and safety and well-designed, well-controlled trials. For coming out with new assays, which can be then directly marketed to consumers and physicians and everyone else, one only has to show that the assay measures what it's claiming to measure, not that it has tests of safety or benefit to patients. And so we've kind of lost an opportunity to help get partners to develop trials that may answer these questions more rapidly about quality control, about heterogeneity in specimens, about what assays are promising, what aren't. And is there any way that we can extricate ourselves from that challenge that we've created? Because I think we do have good collaborative process for developing studies based on therapeutic development. We don't have a good process for developing studies based on these tests unless they're tied to a previously approved drug.
Yeah, and I think you're touching on one of the questions we had in the media materials, which really is whether or not clinical utility should be required for entry of a new test into the marketplace. So I don't know if Liz, if you were able to hear that question, but maybe you have some thoughts about that.
Yeah, so we do see what we think of as clinical utility for the companion diagnostic tests that we approve. What we haven't seen so far is clinical utility for what you might call off-label uses or for tests that don't come through the FDA, namely laboratory-developed tests that are offered under our practice of enforcement discretion. I guess right now we're neutral on that idea that we really think it's important to have an approved test that was used in the clinical trial and actually does have a type of clinical utility established. Whether all physicians have to choose that test or whether they can choose a different test is really outside of our authority. But we do think it's important to have at least one test with clinical utility. Sometimes innovation moves more quickly than regulation. I would say that often happens. So I'm not really sure that there's a specific answer that it's better to have clinical utility for every test before you use it, or whether it's, whether you don't need to worry about it. I just think it depends a lot on what you know.
So, Jim, let me ask you, I think part of the question really was, we have large consortia, cooperative groups, and other types of consortia that for decades have been doing clinical trials of drugs, and yet we don't have a similar infrastructure that has been doing trials of, molecular tests and devices. And is that something that you could imagine that the National Clinical Trials Network would be able to undertake, or some other NCI-supported consortia?
Yeah, well, in theory, yes. I think for this particular area where you're trying to do precision medicine, targeted medicine, whatever you want to call it, it might be better if the consortia that are on the drug development side would actually take on the part of the diagnostic side rather than having it stand separately, because really you can't generate information about the test without having the drug outcome. So adding on that arm would be extraordinarily helpful in my sort of off-the-cuff point of view.
Yeah, I think that's certainly true for companion diagnostics of tests that are used to identify patients to treat with a particular drug. But of course, there are all sorts of other tests that we do to establish prognosis, to monitor response to therapy, and so on, that are not necessarily linked to a particular therapy. And you want to comment?
Yeah, so I don't see that we will have the resources to set up a big network to do that. On the other hand, we have the resources and have initiated a program, because there are actually at least 3, if not more, stages in which the diagnostics have to go through. The first one, of course, is I have 200 samples in my freezer and I come up with a test, and then hopefully I get a validation set from the guy next door. And then there's the end stage, the very, very expensive stage of trying to develop a predictive marker in a prospective, either prospective, retrospective, or whatever the design may be. And then there's the middle of actually bringing the test developed in an academic laboratory up to the appropriate level of quality control that it could even be considered to go forward for study towards approval. And so we actually have a process. We used to call it, RAID for diagnostics. We call it other things now, but there are monies available. We have laboratories, actually some academic and some that are commercial diagnostics companies that we have contracts with to actually help people move their test so it could actually get to the stage where you might want to be able to test it in a large trial.
Thanks, Jim. So I think we'll segue to the next panel, and I think a number of these issues will continue to be discussed in the next panel. So some of us will be staying here, some of us will be replaced by others, and Dr. Trump will be moderating the next panel. Thank you, Neil.